We are sitting at the John Innes Centre, which is a plant and microbial research institute in Norwich, England. We arrived in 1988 and I started my independent research lab in this laboratory. Some of them seem to be quite late. Well, given it's probably long days in here, these are quite late flowering. When I first arrived, I decided to work on a difficult problem, which was how plants knew the season they were in and how they adjusted what they did to that season. And then I went one day to a store because I wanted to buy some tulip bulbs and I had a little pot on my apartment. And the man in the shop said to me, put them in the fridge for 6 weeks before you plant them. And that absolutely intrigued me. Why would I want to do that? And I discovered this process of vernalisation. So this is when plants need a prolonged period of cold before they can flower. If they need winter, how do they perceive winter, register winter? This is what started this whole process of me studying vernalisation for such a long time. And a really big eureka moment was when we realized all of those questions focused on the regulation of one gene. So that was a really big step. So this gene is called Flowering Locus C, FLC. It's a repressor of flowering. So when that gene is expressed, the plant stays vegetative. You can think of FLC as a brake to flowering. When I wake up in the morning, and even in the middle of the night, I'm always thinking about the regulation of this one gene. Now we can actually influence when crop plants flower. For flowering time, we would go in, take the plant, treat it with chemicals and break a particular gene, and then that plant would flower at a different time. So for example, in Asia, they want very, very rapid flowering varieties. Now we can go in and inform the breeders what to actually look for in their populations at the molecular level. So that for lots of reasons, we can actually use our knowledge to help with producing crops. We realized that the memory mechanisms in plants are the same as the memory mechanisms in human cells. And that was another eureka moment in the lab, because it comes down to the DNA in our cells. And this is the science of epigenetics, because it's how the environment influences our own genes. But we don't understand it terribly well yet. So now we can come along and look in the plant system and dissect the molecular basis of epigenetics and then transfer that knowledge to the human system. I'm often asked, how did I get into biology? But I was very intrigued by the documentaries made by Jacques Cousteau when I was a teenager. And he was so passionate about what he did and so beautiful images that I went to university and initially studied marine biology. And one year when I was 18, I persuaded my then boyfriend that we really had to drive to Marseille, all the way from Manchester where I was living, to see if we could meet Jacques Cousteau. When I look back, I think, oh gosh, I was only 18 and I was fairly determined at that point as well.